This is Christian Moreta presenting a just short review for label-free microarray imaging for direct detection of DNA hybridization and single nuclear mismatches. Just a quick background. The DNA microarray is broadly used in different fields such as biology and medicine fields, and it has applications for that, which is for DNA expression profiling and cancer research, as well as drug development and other related to patients survival analysis. And DNA variation sequences affect diseases and development and treatment. And the only way, or one of the ways that it can be seen, is one detect the single nucleotide polymorphisms, or SNP. And through SNP detection, one can give a diagnostic of the patient and see if he's being morphed due to cancer. And diagram just shows a quick workflow of how the process is taken. And the main take here is just to understand they compare two samples and see if the samples are equal, different, and how different they are. And that's how they compare DNAs through this microarray method. Now, the problem established in the paper is that they have a basic way to operate, which is the conventional fluorescence detection. And even although it's a difficult process, it's just a standard process they use. And there's a complication and the intensity to binding energies for the DNA expression, and as well as a complicated procedure, and is always required to be accurately to quantify the genetic expressions, because you want to know exactly the percentage of equivalent between samples. And the way they propose to solve it is using the label-free detection technique, which is remove the need for having fluorescence labeling, and they offer a better quantitative measurement of interactions and eliminate the necessity, obviously, as to use labels, and it's much more faster as well. And this diagram, they trying to express there's a novel method, meaning it's new, and is the DNA hybridization on the microarray. So you can see the upper image compared to the one on the lower is that the lower one has the use of the hybridization, and the spots are darker, and they established using through different experiments by 10, 20 pm, 20 dm, and 18 mm, and different ways to do that. And the diagram on the right, upper right, they show how different they are after and before using hybridization, and that's just established that there is much more better to use it compared to not using it on a specific time. And this second diagram of the benefit is apart from being a good approach to use hybridization, and they show that there's a decrease in the spot while the mass density is increased. But they saw as well that the timing is important too, and how the drop occurs on the mass density. And they established that using their system is a better approach to detect these changes. Now, the result that they obviously kept showing in the work is that there's a single mismatch discrimination, and they successfully show it, and the SNP mismatching, and they show it by using a matched duplex from a duplex containing a mismatch for only one base of pair, and I think it was like a high percentage of a mismatch. And they show in this diagram how different the matches are from A, B, and C, D. This is just that they mismatch, and then D, they show it's really matching example, and they're just trying to clarify this. In this method is much more precise to see the mismatches and the mismatches. Now the conclusion is this to work is this SRIB, which is part of the technology that they develop or the system that they use, is much more useful for this kind of experiment, which is the SNP detection for express analysis, and they use the glass surface, which is it can be applied for microarrays work, and it's much more functional. And one important point is that the system response is not affected by changes in temperature or a refractive index of solution, as we know that we need high temperatures in order to denaturalize the DNA. So it's much easier for us to compare matches and mismatches. So this system supports high temperatures and changes in temperatures, so it's much more stable, and therefore allows melting experiments of both dependent hybridization and denaturalization tests to be conducted. And the next step is, although the signal is just hybridization, it has some noise into it. They offer a solution, but it wasn't too clear how to implement it. And it's at the level that must increase upon hybridization. It's not ideal, so it's much more difficult to see the perfect matches from the mismatches. And they say that they use an iconic concentration temperature of the hybridization buffer and has to be carefully set it in order to solve this problem. Still, it wasn't too clear, and they left some issues and some in the paper. So in my opinion, it needs to be a bit more concise, and it's a bit dense for it to be understood. And that's it.